Imagine trying to understand a bustling, complex city. If you only focused on, say, the criminal underworld, you'd completely miss the architects designing the buildings, the builders constructing them,
Right.
the shopkeepers running businesses, and even those who, maybe unintentionally, create the conditions for certain things to happen, good or bad. You just have a really narrow, incomplete picture, wouldn't you?
Absolutely. You wouldn't grasp the true dynamics, how the city functions, sustains itself, or, what really makes it tick or vulnerable. That intricate web of interactions between everyone, that's what dictates the city's health, its future.
And that's exactly the lens we're bringing to cancer research today. We're not just looking at the rogue cancer cells, but really diving into their immediate surroundings, the neighborhood they live in, the tumor microenvironment. Our focus, a specific group of cells, crucial but often underestimated, fibroblasts. We're digging into this groundbreaking study from Science Advances presenting a huge fibroblast atlas. It's radically refining how we see these cells. So our mission today is to unpack this massive data set, discover the hidden diversity, and figure out the roles these fibroblasts play in cancer.
Yeah, and what makes the study so transformative is how it just shifts our perspective completely. For so long, fibroblasts were kind of treated as this single, uniform thing, like thinking all city workers do exactly the same job.
Right.
But this research, it reveals them as this complex, highly heterogeneous population, each with distinct roles. It's like moving from a blurry photo to this super high-definition map of specialized functions. And that level of detail is absolutely critical for understanding the bigger picture of the disease and, crucially, finding new ways to intervene.
And trust me, you are in for some genuine aha moments today, because this deep dive reveals how these researchers identified an incredible 18 distinct fibroblast subtypes.
18.
Yes, 18. And among them, one totally new discovery, with significant implications for predicting cancer prognosis, even how patients respond to immunotherapy. It's like finding a brand new, super influential profession in our city metaphor, one that seriously impacts everything else. Okay, so before we really immerse ourselves in this atlas, let's maybe just kick things off with the basics. When we talk about fibroblasts, what's the fundamental role people usually think of, and how has that changed so dramatically, especially with cancer?
Well, traditionally, yeah, fibroblasts were known as the builders of our tissues. Their main job is making and maintaining the extracellular matrix.
Okay, extracellular matrix. Sounds a bit technical.
Right, but don't let it sound too intimidating. Just think of it as the scaffolding, the glue, the support network that gives our tissues structure and strength. It's made of proteins like collagen. You probably know that one from skincare ads and other complex molecules. So basically, fibroblasts are vital for tissue homeostasis, keeping things normal, healthy, balanced. They're the diligent construction crew.
The construction crew. Got it.
But our view of these workers has completely changed thanks to new technologies, especially single-cell RNA sequencing, or cis-RNA-seq.
Ah, yes. Single-cell tech.
It's revolutionary, really. It lets scientists look at the gene activity of individual cells one by one. Instead of just getting an average from a whole mix, it's like interviewing every single person in our city metaphor, finding out their exact job, their routine, who they talk to, not just knowing the general demographics.
Wow, okay.
And this has shown us fibroblasts are far from uniform. They show high heterogeneity.
Heterogeneity,
Yeah.
meaning lots of variety, right?
Exactly. Significant variety in their phenotype, their characteristics, their origin, their function, especially within that dynamic tumor microenvironment.
So, yeah, it's like realizing those construction workers aren't just one type. You've got specialized bricklayers, electricians, plumbers, each with unique skills and influences. And when a tumor shows up, some of these specialists might even be, accidentally or actively helping the problem grow. This deep dive isn't just counting them, it's finding out just how diverse their jobs are and how those jobs change from healthy to diseased tissue.
Precisely. The core message here is that truly understanding fibroblasts, good or bad, depends entirely on decoding this complexity, identifying subtypes, tracing origins, mapping where they are, understanding who they interact with. It's a whole new way of looking at it.
Okay, given how complex these cells actually are, I'm genuinely curious. How on earth did they manage to map out such a detailed atlas? This sounds like an absolutely massive undertaking, not just a small lab experiment. Yeah.
Oh, it was monumental, a real testament to collaborative science and advanced data integration. The scale is just groundbreaking. They meticulously integrated data from an astounding 249,156 fibroblasts.
Wow, nearly 250,000.
Yep. 250,000 individual cells, an unprecedented data set for this cell type. And these weren't just from one study, they were carefully collected, harmonized from 73 different studies, a truly comprehensive collection.
73 studies.
And spanning 10 diverse tissues across the body, including a range of sample types: normal, adjacent, tumor, and metastatic.
Okay, can you quickly break down what those different sample types mean again?
Sure. Normal is healthy tissue, no cancer. Adjacent is tissue right next to a tumor, looks healthy, but might show early changes, crucial for understanding the tumor's influence. Tumor samples are the cancer itself, and metastatic samples are from tumors that have spread, indicating more advanced disease. So this really comprehensive approach, pulling from so many sources and contexts, let them build this incredibly broad, high-resolution picture of fibroblast diversity,
Right.
way beyond what one study could ever do.
So it's like you said before, building that global census, detailing every job function across 70-plus countries, 10 industries, and comparing healthy populations versus those in crisis. That's the kind of resolution we're talking about here. It's a staggering feat. Data science, molecular biology, all coming together, really shining a light on this previously murky cellular landscape.
Exactly. And through this massive integration, they provided a high-resolution classification, basically, a detailed ID of 18 distinct fibroblast subtypes.
18. That's amazing.
It's not just finding a few more types. It's uncovering this whole hidden ecosystem of specialized cells, each with carefully defined, shared, and specific characteristics across different tissues. This granular detail lets us move beyond just broad statements and understand the really nuanced roles these cells play.
Okay, so among these 18 subtypes, one really jumped out because it was entirely new to science.
Yeah.
A truly undiscovered player. Let's introduce this star, the TSPAN8 plus chromatin remodeling fibroblast. What makes this one so special? Why did it grab so much attention?
Well, this previously undescribed cell population is, definitely one of the most significant findings. They're distinctly marked by a high expression of TSPAN8, that's a specific protein, part of the tetraspanin family, and other genes like GATHED1, MDM2, and VG's FOFE. But what truly sets them apart and makes them so fascinating is their strong link to histone modification and chromatin remodeling functions.
Okay, histone modification and chromatin remodeling. Sounds pretty technical again. Can you break that down? What does that actually mean these cells do?
Right. Think of our DNA, our genetic instruction manual. It's incredibly long, packed tightly inside cells, not just loose. It's wound around proteins called histones, like thread around tiny spools.
Okay.
This whole DNA histone thing is called chromatin. Now, chromatin remodeling is the process of changing how that DNA is packaged, opening up or closing off access to specific parts of the manual. When a section is open, genes there can be read, activated. When it's closed, those genes are silenced.
Ah.
And histone modification is a key way this happens. Chemical tags on the histones act like little switches, dictating if DNA is accessible or not.
So if other fibroblasts are like the specialized construction workers, building physical structures, these TSPAN8 plus cells are like the master architects who can literally rewrite the blueprints or edit the instruction manual for gene expression.
That's a great analogy, yeah.
They're not just building, they're influencing the fundamental instructions for what other cells become and do. That's a profound level of control.
It really is. These TSPAN8 plus cells potentially have the power to influence the identity, the behavior of surrounding cells by changing which parts of their genetic code are active. They're not just supporting the environment, they're actively shaping it at a genetic level.
That is a game changer.
Yeah.
So these master architects, with their blueprint editing powers, where do they mostly hang out? Are they everywhere or specific districts in the tumor's city? And who are their main collaborators? Who are they influencing?
Good questions. The study carefully validated their existence and location using some pretty advanced techniques. Spatial transcriptome data.
Which maps gene activity geographically in the tissue.
Exactly. And multiplexed immunofluorescence.
Yeah.
Which is like taking a super detailed multicolor photo showing different cells and proteins and how close they are.
Right.
What they found was fascinating. These TSPAN8 plus fibroblasts were primarily found in the tumor margin area across various cancer types.
The margin. So the edge where the tumor is growing, invading.
Precisely, right at the leading edge where the action is. And crucially, they showed a strong tendency to colocalize, meaning they're found right next to endothelial cells.
The blood vessel lining cells. Okay.
And their key interactions really highlight their influence. They mainly talk to those endothelial cells using a signal called VGFA, vascular endothelial growth factor A. VGFA is famous, or infamous really, for promoting angiogenesis. new blood vessel formation.
Which tumors desperately need to grow and spread.
Exactly. So a cell pumping out VEGF-A at the tumor margin is a big deal. They also interact with T cells, critical immune cells, via a receptor called F2R. So they're influencing blood supply and potentially immune responses.
Okay, it definitely sounds like these TSPN-A plus cells are highly active, equipped with some powerful tools, reshaping the environment fundamentally. But what does this actually mean for a patient's journey? Is this good news, bad news?
Unfortunately, the findings here are, well, pretty concerning. They point to a troubling association. The analysis clearly showed that the presence of these TSPN-A plus chromatin remodeling fibroblasts was consistently associated with poor prognosis.
Prognosis, the likely outcome of the disease.
Right. How likely recovery is. In fact, a high score for these cells, more of them, or high activity, was significantly linked to reduced overall survival in several aggressive cancers, including lower-grade glioma, LGG, kidney papillary cell carcinoma, copper, mesothelioma, sarcoma, SARC, ovarian cancer, and stomach adenocarcinoma, statin.
Reduced overall survival.
Yeah.
So patients live shorter lives after diagnosis if these cells are abundant.
That's what the data strongly suggests.
So if these master architects are numerous and active, rewriting blueprints, boosting blood vessel growth in the tumor's expanding city, the outlook is significantly worse. They're basically constructing conditions that are bad for long-term survival. That's a pretty stark connection for a newly discovered cell.
It absolutely is, and it leads right to the next critical question. What about their role with modern treatments, specifically immunotherapy?
Ah, right. The treatment's harnessing the immune system.
Exactly. The study found that higher TSPN-A plus fibroblast scores were significantly associated with a poorer response to immune checkpoint inhibitors, ICIs, instead stomach cancer.
Okay, quick reminder. Immune checkpoint inhibitors.
Sure. They're a type of immunotherapy drug. They work by blocking certain brakes on immune cells, especially T cells, basically letting the immune system attack cancer more effectively.
Got it. So poor response to these cutting-edge drugs is not good.
Not good at all. Highly significant. While the results varied a bit in other cancers, this finding strongly suggests these TSPN-8 plus cells might contribute to treatment resistance. Perhaps by actively modulating T cells through that F2R interaction we mentioned, kind of dampening the immune attack or making it less effective.
Wow. So a cell type we didn't even know existed could be a critical predictor for how well patients respond to some of our most advanced treatments. It just underscores the deep, intricate connections. It's not just the tumor cells. The whole neighborhood dictates the outcome in response to therapy. So this atlas wasn't just about finding this one new cell type. It painted a much richer picture of all fibroblasts. What else did this huge mapping effort tell us? About their broader distribution, how they differentiate, become specialized, maybe even how they age in the tumor context.
Yeah, it revealed really fascinating distribution patterns, emphasizing how widespread certain subtypes are in cancer. For instance, 6 fibroblast subtypes, including our TSPN-8 plus star, plus others like MMP11 plus myofibroblasts, CFD plus inflammatory fibroblasts, showed significantly higher proportions in tumor samples across tissues.
Meaning they're pretty much everywhere in tumors.
Ubiquitous, yes. Key players in cancer progression, seemingly regardless of where the tumor started. Other subtypes tended to be more tissue-specific, found more often in particular organs. They also looked into origins and differentiation pathways using trajectory analysis.
That computational mapping of how cells might change.
Right. And intriguingly, TSPN-8 plus chromatin remodeling fibroblasts were found to largely dominate the root of tissue-resident fibroblasts, suggesting they might be a kind of foundational, maybe almost stem-like cell type that other specialized fibroblasts can differentiate from.
The original blueprint holders, maybe, from which other specialized architects emerge.
That's one way to think about it, yeah. Constantly adapting.
So it's not just what they are, but what they become. How about aging? Does the age of these cells matter?
Critical question. They looked at cellular senescence scores measuring cellular aging. Senescent cells stop dividing but stay active, secreting stuff that influences their surroundings, often promoting inflammation. What they found was striking. Fibroblasts generally had the highest senescence scores compared to other cells in the tumor microenvironment, and these scores increased going from normal to adjacent, tumor, and metastatic samples, indicating fibroblasts tend to age faster as cancer progresses in a tissue.
That makes sense.
Yeah.
Any twists?
Oh, yeah. A really complex, intriguing finding with the TSPN-8 plus fibroblasts specifically. They showed a significant negative correlation between senescence and aging in the ovary and colon.
Negative correlation.
Yeah.
So Less senescence with age.
Exactly. Counterintuitive. In those specific tissues, as the patient gets older, the TSPan8 plus fibroblasts actually showed less senescence, not more. They seem to stay more youthful, more active.
Huh. Why would that be?
We don't fully know yet. It really highlights how tissue-specific this is, different mechanisms in different parts of the body. But for you, the listener, think about it. It implies that in certain cancers, these detrimental TSPan8 plus cells might keep doing their disease-promoting thing for longer, maybe contributing to more aggressive cancers in those organs. It's a complex layer we're just starting to figure out.
That is a surprising twist.
Mm-hmm.
Not just older cells, more aging. It shows how nuanced this all is. And finally, beyond TSPan8 plus hair, what about the wider social network? Who are all these diverse fibroblasts talking to most?
Well, the atlas mapped out this cellular communication network. Beyond TSPan8 plus 8 fibroblasts interact most often with other fibroblasts.
Talking amongst themselves.
A lot, yeah. Coordinating. After that, significant interactions with endothelial cells, blood vessel liners, and epithelial cells, organ liners. Interestingly, less interaction with immune cells generally, though more with macrophages compared to T cells and B cells. But take another example. The CFD plus inflammatory fibroblasts, another subtype found everywhere in tumors. These guys play a big role in immunomodulation, influencing the immune response. They interact with T cells, B cells, and macrophages through a specific pathway, CXCL12, CXCR4.
So they're actively talking to immune cells, potentially shaping how the immune system responds or fails to respond.
Exactly. It's this constant dynamic cellular conversation, directly impacting the disease outcome. Hashtag tag outro.
So what does this all really mean? This deep dive into the fibroblast atlas just fundamentally changes how we see these support cells. By pulling together this massive amount of data, researchers didn't just classify 18 fibroblast subtypes, which is huge in itself, but they also uncovered the critical TSPan8 plus chromatin remodeling fibroblasts, a previously unknown player that significantly influences cancer prognosis and even immunotherapy response. This isn't just adding a little knowledge, it truly expands our view of the complex tumor microenvironment, reveals a whole new cast of characters with vital, often detrimental, roles.
Absolutely. And the insights, particularly about TSPan8 plus unique epigenetic role, that blueprint rewriting ability, their location, their interactions, it paves the way for incredibly exciting future research. This could fundamentally shift how we approach treatment, maybe lead to new biomarkers to predict disease or treatment response, or even more powerfully, maybe new, more targeted therapies, not just attacking cancer cells, but disarming their fibroblast allies, or even, reprogramming them to work against the tumor.
Imagine that, turning a villain into an unexpected hero.
It's a tantalizing possibility.
It really makes you wonder, doesn't it? If zooming in on just one previously unknown cell type like TSPan8+ reveals such powerful insights, what other hidden heroes or silent villains are still out there waiting to be discovered in our own bodies? It's just a profound reminder that the microscopic world holds macroscopic answers. There's so much more to learn about this intricate cellular ballet happening inside us all the time.
And that leads to an important question for you, our listener, to think about. Given this incredible diversity of cells, their complex interactions we've just discussed, what do you think is the single biggest challenge researchers face when trying to translate these amazing cellular discoveries from an atlas like this into